Hello everyone, this video is about biochemistry practical sessions. I won't be, conducting the practical demonstrations. We are just reading the material, reading the study material of this biochemistry practical sessions, theory materials. So let's start. Let's have an introduction about this. Biochemistry is a branch of biology that is defined as the chemistry of living things. It deals with the structure of chemical compounds that make up living system. The transformation of these chemical compounds and the physiochemical processes constitute the basis of our vital activity. The biological phenomenon are analyzed in terms of biochemistry. For example, one is to see and read things only through the chemical reactions occurring in the eye and brain effectively. You are aren't able to taste the lunch through the chemical reactions in the taste buds. Kidney collects and processes urine through the elaborate mechanism. This clearly shows that activities taking place inside the body are as a result of chemical reactions. Hence, biochemistry is also called biological chemistry or chemical biology. This deals with organic compounds. So, biochemical aspects of this biochemistry is said to be a offshoot of organic chemistry. The term biochemistry was proposed in 1903. Distin von Liebig was known as father of agricultural biochemistry. So, biochemistry and physiology. Biochemistry is an offshoot of human physiology, formerly called physiology. Now, these two are separated. Physiology deals with the chemical aspects of plants and animals and their functions. Branches of biochemistry. Biochemistry has two branches, namely descriptive biochemistry and dynamic biochemistry. Former deals with qualitative and quantitative characteristics of various cell components or compounds. Then dynamic biochemistry deals with the elucidation of nature and mechanism of reactions involving the cell components. And scope of biochemistry. Advancement in biochemistry have found large scale application in various areas of industry, agriculture, medicine and pharmacy. Then genetic engineering. By applying biochemical techniques, improved strain of plants and animals are produced through genetic engineering. Nitrogen fixing genes of bacteria are transferred to higher plants, wheat and maize, for example. Then food and proteins. New strains of microorganisms are being employed in the production of low cost food proteins and essential amino acids for domestic animals. And biological control to protect cultivated plants from pests. Biological preparation of superior quality that is not harmful to humans and animals are currently being manufactured. And control of pollution. Biological methods for dispersal and recycling of industrial and domestic wastes and for cleaning common um, condiments and of seed and usually cultivated bacterial mutants have proven to be sustainable value, having a sustainable value. And industrial development. Biochemical processes are widely used in the food industry in the preparation of bread, cheese, cream, and in leather industry, along with the detergents with enzyme addictions to be are also available. Next include the medical field. Biochemical methods are increasing by gaining acceptance in the pharmaceutical field. Enzymes are used in technology for the synthesis of pharmaceuticals such as insulin, vitamins, antibodies, antibiotics, amino acids, nucleotides, and nucleosides. So, first experiment is about actual and titratable acidity of strong and weak acids. Actual and titratable acidity of strong and weak acids. Our aim include to determine the actual acidity and titratable acidity of a strong acid and a strong and weak acid along with that. principle, acids when dissolved in water dissociate to give protons and the corresponding ions. The strength of an acid depends on its capacity to dissociate. A strong acid like HCl and sodium, in solution dissociate easily. A weak acid like CH3COH has poor tendency to dissociate. Thus, it will contribute to less quantities of H plus ions compared to HCl of the same molarity, or it can be tested by checking the H plus ion concentration of solution by 0.01 normal HCl and 0.01 normal CH3COH with Chapfer's indicator, I'm not sure about the pronunciation, which gives a yellow color in the vicinity of H plus concentration of 10 raised to minus 4 m pH 4, and the minus 4 is a red color at concentration of 10 raised to minus 3 m or above. So HCl gives H plus plus Cl minus, and CH3COH undergo dissociation to form CH3CO minus plus H plus. H plus Cl minus. Then CH3COOH gives CH3COO minus plus H plus. However, strong and weak acids have the same tritable acidity when titrated against alkali with phenolphthalein as indicated. This is because the addition of alkali to a weak acid like CH3COOH neutralizes H plus and pulls the reaction towards nitric acid right, and complete dissociation is affected at the end point of titration. Then this is because the addition of however, strong and weak acids have the same titratable acidity when titrated against alkali with phenolphthalein as indicated. This is because the addition of alkali to a weak acid like CH3COH neutralize H plus and pulls the reaction towards right in the sense, just to get this equation, CH3CO minus plus H plus direction, and complete dissociation is affected at the end point of titration. Okay, and materials required, we need 0.01 normal HCl, along with that 0.01 normal CH3COH, then 0.01 normal NaOH, phenolphthalein as indicator. So these are the results which we later we are going to learn, and this Jaffer's indicator also. Then procedure, take about 2 ml each of CH3COH and HCl solutions in 2 test tubes. Take about 2 ml each of CH3COH and HCl solutions. We are going to compare this acid. So we need both of them. So both of these acids, not them, both of these acids. Take about 2 ml each of CH3COH and HCl solutions in 2 test tubes. Add 2 drops of Jaffer's indicator each and note the color. Then pipette out 10 ml of 0.01 normal CH3COH in a 100 ml conical flask. Add 4 drops of phenolphthalein. Then indicator and titrate. The usual procedure of titration. Either you can just follow double burette method or single burette method. Here we are mentioning about the single burette method. Just mouth pipetting is also involved in this. Mouth pipetting, or either you can use a bulb pipette, whatever it may be. That method we are going to discuss. So 4 drops of phenolphthalein indicator and titrate against 0.01 normal NaOH until a pale pink color is indicated or obtained. Repeat the titration with 10 ml of 0.01 normal HCl. Compare the titration values. So we got a result which represented with this column. So just try to interpret this. We got volume of HCl pipetted, the amount we are taking in a conical flask to titrate against the burette reading. 10 ml burette reading final, written like this. 1 ml NaOH is used, but same is the amount used for CH3COH also. 1 ml of NaOH is used. That means there is a relation which we are going to interpret as an inference. Okay, So acids when dissolved in water dissociate to give protons and corresponding ions, conjugate bases. The strength of an acid depends on its capacity to dissociate. A strong acid, HCl in solution, easily dissociate completely into H plus and Cl minus ions. While CH3COH, which is a weak acid, will dissociate into CH3CO minus and H plus ions. The CH3COH will contribute only less H plus ions when compared to HCl of the same molarity, but HCl require the same NaOH for the reaction being indicated by the appearance of a pale pink color, that is the endpoint color. Okay, so HCl and NaOH has same titratable acidity when titrated against a strong base like NaOH, because the addition of NaOH to weak acids neutralize H plus ions and pulls the reaction to forward, and complete dissociation is attained at the endpoint of titration. And titer value in relation to the titer acidity of strong acid 1 ml, titer value of in addition to the titer acid of a weak acid CH3COH both 1 ml. By using Schoffer's solution, we observed variable acidity for strong acid and weak acid, but the titratable acidity for both weak and strong acids are same. Variable acidity for these two acids, one is strong, one is weak, but their titratable acidity is equal. Okay. Then next, comparison of the buffering capacities of two buffers of same pH. A buffer is a system consisting of an acid and its conjugate base that resists changes in hydrogen ion concentration. An acid or alkali is added. So smaller the changes in pH, greater is the buffering capacity. The buffering action is maximum around the pH value of acids and extends roughly over 1 pH unit on either side of this value. These various buffers will have differing range of buffering action depending on the pK values of conjugate groups of acids. pK is the negative logarithm of the dissociation constant of a reaction. So the carboxyl group in the CH3COH has a peak value of 4.75. Acetate buffer will be effective in the pH range 3.75 to 5.75. And the dihydrogen phosphate H2PO4 has a pK value of 6.8, and phosphate buffers will be effective in the range 5.78 to 7.8. So dissociation is also shown. CH3COH gives CH3CO minus plus H plus. And H2PO4, HPO4 2 minus plus H plus. Acetate buffers of pH 5.7 will not be very effective in resisting pH change. Then alkali is added to it, unlike the phosphate buffer of the same pH. On the other hand, the acetate buffer will be effectively resisting the change in pH when an acid is added to it. Whereas the phosphate buffer will not be too effective. This is easily predicted in the amount of acid or alkali needed to change the pH of acetate buffer 5.7 to 3.5 and 8.5 respectively, and comparing the values obtained in the case of phosphate buffers 5.7 in similar experiment. It is worthwhile to note that when pH changes from 5.7 to 3.5, H plus concentration increases 159 fold from this value as shown here. Similarly, when pH changes from 5.7 to 8.5, H plus concentration decreases from this value to another one. So procedure include pipette 10 ml of acetate buffer into 100 ml conical flask. Add 5 drops of phenolphthalein solution and titrate it against 0.01 normal NaOH to a pale pink endpoint. Note the titer value. Repeat the titration with the phosphate buffer. Titrate the buffers similarly against 0.01 normal HCl with Joffer's indicator and to the orange endpoint. If necessary, use a control solution for color comparison. Then note the titer value. Compare the titration values and assess the capacity of the two buffers. So reagents, we mentioned about 0.01 normal molar acetate buffer 5.7 pH. Then 50 ml of 0.02 m acetic acid, add 0.1 NaOH. Drop by drop until the pH is 5.7 as indicated in pH meter. Dilute it into 100 ml. Then 0.01 normal NaOH along with that 0.01 molar phosphate buffer to 50 ml of 0.02 molar KH2PO4. Add 0.1 molar NaOH until the pH is 5.7. Dilute it into 100 ml. And 0.1 normal HCl, phenolphthalein indicator and Joffer's indicator. These are required. And we mentioned about the procedure too, how to do this. And another major point is about reactions of monosaccharides, disaccharides and polysaccharides, and along with that lipid we will be learning. All of this are very important, so which will be included with the help of next video. Thank you for listening.